Welcome back to the next part. So we now have something more interactive. How to interpret clinical trials, and I have two panelists, Dr. Savarese and Dr. Lewis. So this is really about showing trials and discussing principles of how to interpret the results of trials. Okay, I would like to start with this. So 3 questions you have to ask yourself if you look at a clinical trial. Why was it done? Who was studied and how was it done? So 3 simple questions. So if we start like this, the why is the hypothesis of the trial. So we picked the IMPROVE-IT trial. And that was a big study in ACS patients investigating the value of ezetimibe in lowering cholesterol and events in patients post-ACS. So 3 questions, 2 of them I would like you to remember. Number 1, does lowering LDL cholesterol with a non-statin agent, ezetimibe, reduce events? So question number 1, keep that in mind. And number 2 is even lower, even better, which means bringing down cholesterol lower than the recommended target value of 70. Does that reduce more events? So is even lower, even better? 2 questions, keep that in mind. Okay, who was studied? Inclusion criteria, so you see patients with ACS and patients with lowish LDL cholesterol. So patients had to have LDL cholesterol, let's say around 100 to be included. How was it done? That's the study design, so that's very important when you look at interpreting a trial. So how was it done? So we had patients post-ACS 10 days. And you can see LDL cholesterol between 50 and 125. This was inclusion criteria. And then 18,000 patients were randomized to receive either simvastatin, 40 milligrams, or simvastatin plus ezetimibe. So that was the treatment arm. And the goal was to achieve certain LDL cholesterol levels. So there were follow-up visits, and the duration was not set by year. It was an event-driven trial. The plan was 3 years. Approximately it took 7, okay, because the event rate was lower than expected. Then there was a primary combined endpoint. This is very important to keep in mind what the primary endpoint is. And you see it's the CV death, MI, and then hospital admission for unstable angina, coronary revascularization or stroke. So if you like, that's a MACE endpoint plus hospitalization and revascularization, so a combined endpoint. Baseline characteristics, always look at that. You see typical characteristics here, 1/4 females, 1/3 diabetics. You can see here different types of acute coronary syndrome, so that's quite classical, I would say. And that is something important here. That's the mean LDL cholesterol at enrollment. This was 95 milligrams per deciliter, so kind of low. All right, so this is what was achieved, LDL cholesterol changes, and if you look at the medium time average LDL, it's approximately 70 in the Simva arm and approximately 53 in the ezetimibe arm. So 70 was reaching the goal that was set by guidelines, and the other one was approximately 17 milligrams per deciliter lower. This is something to be kept in mind when we interpret the trial as well. So, and now this is the primary endpoint in the intention to treat analysis. Again, this combined endpoint. And you see the Kaplan-Meier curves here. And when you look at this, there was a relative risk reduction of 6% over 7 years. Statistically significant, number needed to treat of 50 over the course of 7 years. So these are the results. Now, what I would like you to tell me is, what do you think does these results mean? Okay, so remember the questions, the hypotheses of the trial. So have we achieved these hypotheses? Yes? Yes?
At least one of them.
Which one? Sorry? No, just say it again.
You answered the first question. I mean, you can't say so much about this lower the better or something.
Okay. So you say somewhat achieved, okay?
If I remember, memory serves me right, say non-statin.
Exactly. So can we... So question number 1 is even lower, even better. And the question number 2, can we achieve this with a non-statin agent, with ezetimibe? Well, what is it? The answer is yes, right? So you have, it's of course on top of statin treatment, but we had simvastatin in both arms, and on top there was ezetimibe. We do have a significant reduction of events. So yes, so ezetimibe can reduce cardiovascular events. Question number 1 is even lower, even better. We have 70 milligrams per deciliter target, and we have 53, so that is lower, and it's significant, right? So if you look at it that way, if you interpret it, you can say, yes, the trial has achieved what it wanted. So the research questions were answered. So the next thing is, of course, how do you interpret that in the clinical setting? So, you know, do we think that in the clinical setting, when we make treatment decisions, does everybody need ezetimibe? Yes, no. This is a completely different question, and I don't want to go into this. I would like to show this. This was very nicely done, I think, when you look at this regression curves showing or investigating the reduction in LDL cholesterol and the reduction in event rates that we have in the different lipid trials. You can see here it's perfectly on this line. So for the 17 milligrams per deciliter LDL cholesterol reduction, there was the number of events reduced were perfectly on this line that were thought that we could achieve. Okay, so we have just a low reduction in LDL cholesterol, but this translates perfectly into reduction in events, and it fits perfectly to what we know from other trials. Okay, should we give this drug to certain patients? That's a different question, and I don't want to ask that question now. Okay. All right, so now we have primary and secondary endpoints. So again, the primary endpoint here is a combined endpoint, statistically significant. Then there are other secondary endpoints, also combined endpoints. My point here is, if you look at them, somebody might go and say, hey, great, secondary endpoints all positive. So my questions, are these secondary endpoints valid? Can we say those are significantly reduced? What do you think? Yes? Okay. What is the important fact? It could be, let's say, primary endpoint non-significant, secondary endpoint significant. We do have that sometimes, right? So let's say we have a combined endpoint not significant, MACE endpoint, secondary endpoint significant. It is consistent. Completely agree. So the primary endpoint, the power was made for the primary end. The study was made for the primary endpoint is significant. Then we're allowed to test further for secondary endpoints. They are significantly reduced, so that's valid. Okay. But keep in mind when the primary endpoint has not been reached, well, it's not statistically significantly different, then it doesn't really matter what is beyond that. That's hypothesis generating. Okay? All right. So levels of endpoints. I like this one. The best endpoint is this. That is the hardest endpoint we have, to count dead bodies. You know, heart failure trials, we can count dead bodies. So all-cause mortality. Then it's cause-specific mortality, for example, fatal myocardial infarction, cardiovascular death. And then there's non-fatal clinical events, non-fatal MI, non-fatal stroke, hospitalization. These are quite important too, also for health economics, obviously. And then there's surrogates, LDL cholesterol, HbA1c, whatever you want to take. And we always got to be careful with that. Then there's quality of life. I'm not sure if it should be that low. I think quality of life is quite an important thing. I think it should be higher. But this is important. This is just a surrogate. And we know that not all drugs that lower LDL cholesterol actually lower events, right? Let's take niacin, for example, and nicotinic acid and stuff like this. Okay, when we look at the endpoints of the IMPROVE-IT trial, all-cause mortality not different, cardiovascular death not significant, but then we have two here, MI, stroke, ischemic stroke. When you look at this, so here's a significant reduction. Oops, sorry. So are these important individual endpoints? I think they are. That's non-fatal events. Okay, subgroup analyses. That's the rule of the 4 Ps. I like that one. So the subgroup analysis needs to be pre-specified, adequately powered, plausible, and practically relevant. You can test anything in subgroup analysis, but does it make sense? Maybe not. I think you heard something about that before. Okay, what about improve it? Just look at this. These were pre-specified subgroups of the trial: sex, age, diabetic status, prior lipid-lowering therapy, yes, no, and the baseline LDL cholesterol. So all of these, I think, they fulfill the 4P rule. And now look at this. You see the point estimates here and the confidence intervals. And here is one with an asterisk, which has a P for interaction of 0.023. So when you look at this, or maybe I ask my question first. So what do you think of this subgroup analysis? So what does it mean? We have this for the diabetics. If we go back, this looks not so bad either. What do you think of this one here? You know that people might come, maybe pharmaceutical industry might come and show you this and say, hey, we have special benefit in this group, you know, with prior lipid-lowering therapy. What do you have to look for? I put it here. Look for the P for interaction. Okay? So this has a significant P for interaction. So this is a subgroup where there is some difference. The others, no. There is no difference. What does it mean? The results of the overall population can be attributed to the subgroup. Okay, this is what it means. There might be a difference here in the diabetics. But so check out the P for interaction. That's important. Okay, so now what they did a subgroup analysis in diabetic and non-diabetic cohort. So when you look at this, these are the diabetics. They have higher risk. You can read this with a Kaplan-Meier risk estimate here. And you see a difference, relative risk reduction of 14% in this group. And then you have the non-diabetics where there's no difference. So now again, what does that mean now? Should we only treat diabetics now? No? Why not? Very good. I agree. Why? Yeah, perfect. Perfect. Absolutely. Absolutely, I agree. Okay, the trial was done in the overall cohort, right? And we have results for this. So we should treat all of them. Completely right. But it does show that, number one, diabetics are at higher risk, and maybe they benefit even more, but it's completely right. If you want to know, you need to do a new trial and test this hypothesis. So hypothesis generating. Excellent. Good. Very good. So we switch to another topic. This is anticoagulation for atrial fibrillation. So this is the ROCKET trial, which tested rivaroxaban versus warfarin in patients with atrial fibrillation at increased risk. This was the statistical methodology. So you see something about sample size, primary efficacy evaluation. The primary efficacy parameter was stroke and non-systemic embolism. And you see here non-inferiority on treatment, superiority on treatment, testing, and then intention to treat. Okay? So these are the results. Kaplan-Meier curves separate quite early. Non-inferiority met quite clearly. And now when we look at the next, this is now testing for superiority. Now you have the on-treatment, oops, how did I do that? So now you have the on-treatment cohort, and you see here, significant p-value, and you have the intention to treat, not significant. So you can imagine that that makes quite a difference, especially for the pharmaceutical company that sells the drug. So what should we do? Intention to treat or on-treatment analysis for testing superiority? Intention to treat? On treatment, intention to treat? Okay. So I think Alexander talked about that. Okay, so it's intention to treat. So intention to treat is the gold standard to test for superiority. Obviously, the company liked this here very much. Okay, and you know that there were some difficulties. Many events occurred after active treatment in the follow-up period after that. But that is important. So just to show you this, that's the full study period that is covered in the intention to treat analysis versus the on-treatment period, which is always active treatment plus some extra time, which is defined in the study protocol. So if you have an event, a stroke, outside the on-treatment period, it's not counted in the on-treatment analysis, but it is counted in the intention to treat analysis. So it gives you a better idea. That's like, how can I say that? The analysis that is always assuming the worst case, and this is why we're using intention to treat for this. And you can see in the on-treatment analysis, we count this. Sorry, in the on-treatment analysis, we do not count this event. In the intention to treat, we do count this event, and that does make a big difference. So the basic rule is if you test for non-inferiority, and Alexander told you about this, it's on-treatment analysis. When we check for superiority, it's intention to treat, and safety is always the on-treatment analysis. I think he touched that as well. Okay, these are the standards. Okay. Now the Aristotle trial. I have a different point to make here. This is also anticoagulation, same thing, testing, in this case, apixaban versus warfarin in patients with atrial fib. And these patients received 5 milligrams BID, apixaban or warfarin, and there was a reduced dose, like in all of these trials, which is a 2.5. And as you may remember, the 2.5 milligram dose was only given to patients with certain characteristics. For example, impaired kidney function, or the 203 rule, which involved age, body weight, and kidney function. So pharmacological criteria. Okay, so statistics, very clearly, primary outcome was the same, stroke systemic embolism, non-inferiority. After that, tested for superiority, bleeding, for superiority, and then it was all-cause mortality that was tested. These are the findings. So non-inferiority met, superiority met, so apixaban was superior to warfarin in preventing strokes and systemic embolism. These are the safety events. So now I'm just going back to this. Now you've seen the results. So a number of, I think, like Russell, 5-6% of patients had the 2.5 dose. So now my question for you is when we have the trial results here, what do we know about this dose of 2.5 milligrams? The reason I put this here is how can we interpret the results for the patients treated with 2.5 milligrams? The reason for this is that we see many patients treated with a lower dose who are not actually adequate for this dose. Okay, it's because doctors don't want to have bleedings and they go down to the lower dose. So what do we know? Do we know anything about the 2.5 milligrams? I'll just show you this subgroup analysis. Okay, result for all patients. This is stroke, this is major bleeding, this is the all patient, and this is the 2.5 milligram dose, and this is the 5 milligram dose. P for interaction not significant, which means both groups do identical, right? So can we treat all patients with 2.5 now? No, why not? Yeah, of course, exactly. And that's very, very important. That's the point I would like to make. So there were very clear criteria when to use the reduced dose. That is mere pharmacology. So it's according to kidney function, age, and stuff, which of course translates into different plasma levels. Okay, so basically in patients that are older, have less weight and less kidney function, plasma levels will be higher. So it's a pharmacological definition. And only in those patients we can say that the 2.5 milligram dose is as good as the 5 milligram dose in the other patients. Okay, but if you treat a patient without these criteria with 2.5 milligrams, you might have underdosing, and you cannot say if you have these results. And that's important, and physicians don't understand this, and they always, not always, but many, I think it's like, 30-40% that are used where the lower dose is used, although it should not be. And we know that this can be hazardous and increased stroke rate. Yes, but you had a question.
Yes, what about with triple therapy?
Triple therapy, oh, that's a completely different story. We do not have data on this yet. We will have with the Augustus trial. I showed you the Augustus trial yesterday. So the Augustus trial is exactly testing this, but it's only investigating the 5 milligram dose. But then 5 milligrams of apixaban versus warfarin, and then another arm testing triple versus dual therapy. I showed you that yesterday. Okay, we have no data on 2.5. And I'm not very happy with 2.5 in triple therapy because it's half dose. It's different with dabigatran and rivaroxaban. Okay, but the point I would like to make is you need to be sure that dosing is according to the criteria that we have, and that's true for all of these drugs. Okay, now this is the ENGAGE trial. This is edoxaban in atrial fibrillation. And it's again a big trial, again patients with AF at increased risk. And here we had 3 treatment arms. It was warfarin, it was high-dose edoxaban 60 QD, and low-dose edoxaban 30 QD. And then again, there were dose reduction criteria also in this trial. Primary efficacy endpoint, stroke and systemic embolism. So why am I showing you this? Because they used a different methodology. So they said primary efficacy for non-inferiority was on treatment, and they used something modified intention to treat. Okay, and superiority testing was intention to treat analysis. Safety, again, on treatment. So what is modified intention to treat? Does anybody know what that is? I'm not sure if Alexander touched that. Okay, what is intention to treat? The patient is randomized, right? And he's in the trial, regardless of whether or not he takes the study drug, right? So what is modified? I think it's quite a smart idea. We had this example of the guy who was randomized and is run over by a bus, right? You remember that? Without having ever taken a drug, and he's counted as an event. Okay, this person who's randomized, run over by a bus, that's an SAE, that's an event. So, okay, so what does it mean? Modified intention to treat are all patients randomized who took at least 1 dose of study medication. That's what they do. Just to show you, it did not make much of a difference in this trial. So when you look at this, we have, let's say in this group, we have 7,034 patients, and in the modified intention to treat, 7,002. So 30 patients were actually randomized and did not take a pill. So they were not included in the modified intention to treat. Okay, so why should we use this? And I asked this to Basel. That's a good question. I cannot answer this. We thought that they wanted to be even more correct. Yeah. Okay. I'm not sure if that will be the standard for other trials. I think it's still intention to treat analysis, which is the standard for superiority testing. And I would like to show you that again in this trial it did make a difference. Sorry? Yeah. So just to show you here, what they did was they did the on-treatment analysis. This is for the primary endpoint. And look at this. It says p-value for superiority. It's significant. That's in the on-treatment analysis. And when you look in the intention to treat analysis, superiority was not met. Okay, so again, it made a difference for interpreting the results. Okay. So now this.
What did they conclude from those results?
That it's non-inferior. That's what you have to conclude. These are the statistics. So honestly, I do not know why they chose the modified intention to treat and put this in this analysis, because in the statistical plan, they said in the end, superiority testing is made by intention to treat. So honestly, I can't tell you why they did it. But the interpretation is clear, it's non-inferior. So that's what they said. Okay, so right after that, there was this meta-analysis. So with the results of the ENGAGE trial, there was this meta-analysis published in The Lancet, NOAC treatment versus warfarin treatment in patients with atrial fibrillation. You see it here. This is for stroke systemic embolism. This is for major bleedings. This is for secondary endpoints. So different drugs, different doses, different patients maybe. So what's the value? Is it adequate? Does it add value? Is it correct? What do you think? Look at this. Looks quite good. Significant. Sorry?
Anyway, both has high heterogeneity. 57%, 53%.
So why is it valid to do this? Or is it not? It's completely right. You have to check the heterogeneity, and you can see here it is quite high but not significant. That's for the primary efficacy endpoint. when you look at bleeding, look at this. Okay, it looks completely different. Why is that? What do you think? You talked about this? Okay,
Yes.
all right.
In a different.
It's because of definitions of bleeding, right? So that's why it has more heterogeneity. Okay, but why can we use this? Why is it, let's say, technically okay to do this meta-analysis? I know Gianluigi's answer to this. What is it testing? It's not testing drug versus drug, right? It's testing a principle. It's testing. Yeah, and it's testing the principle of NOAC versus Warfarin treatment. And I personally think it's a good analysis. I think it does add some value. But you always have to check out, when you see this, you have to check out the heterogeneity. I think that's quite important. Yeah, Basel.
I think it's another thing that people even missed when they were writing the paper. Basically, well, they didn't miss it. They knew it. But what you said is absolutely right. It's a principle of the new treatment versus the old treatment. Because remember that the NOACs are different, totally different drugs. One's a 2A inhibitor of the bigger train, does totally different things to engagement to 10A. And so you can't really compare the bigotran and Pixaban as John mentioned us, is that if you're thinking in terms of mechanisms,
right?
Pharmaceutical mechanisms.
Yeah.
But you can say a new way.
A new principle has been tested. Yes, absolutely.
I completely agree with you. It's Warfarin versus non-Warfarin.
Exactly, exactly. Okay, great. So I have something different now. Now this is on lipid treatment, evolocumab. It's the OSLER program. So you might be aware of that. So the OSLER program was a very heterogeneous trial program, as I can say. So evolocumab was tested in all kinds of different patients populations in phase 2 and phase 3 studies. Now what they did was the following. They had all these phase 2 and phase 3 studies, and those patients who were on evolocumab and were free of side effects and took their pill were asked to take part in the OSLER program. Okay? So in the end, 4,465 patients of the previous phase 2, phase 3 studies were enrolled in the OSLER program and were then randomized. So that was like a new study cohort, and they were randomized to receive evolocumab or standard of care. Okay? And then there were like OSLER 1, OSLER 2, which different cohorts because they took the phase 2 trial patients and on the other hand, the phase 3 trial patients. And then the data from both trials, if you wish, were combined in the analysis. Okay, so now here's my question. Do you think that's okay? What do you think about the study design? Is that adequate? What do you think? It's important, isn't it? I mean, that was very, very major information coming out for the new drugs. So it did make a difference. So is it okay?
Will potentially be some selection bias if you ask participants
Of course.
from the first study
Of course.
versus the next?
Of course. Okay, number one, I have to say OSLER was a safety study. Okay? It's not an outcome study, it's a safety study. But you're completely right. Of course it's selection bias because you're only taking the patient who actually were on drug and tolerated the drug. I personally think it's a very, very smart idea to have very low dropout rates potentially in the trial that's coming. So I think it's correct. And from a statistical point, it's completely correct. But you're right. It selected certain patients. So, but they wanted to do this because this was the intention of the trial. OSLER was testing primarily safety, secondarily LDL lowering, and then they had an exploratory outcome, a pre-specified outcome on events. Okay, so primary safety, secondary LDL, and then an exploratory outcome, which was cardiovascular events. So just very briefly, this was testing the safety, a little signal here with neurocognitive events. Secondary endpoint LDL cholesterol reduction, about 60% relative reduction in these groups. And then look at this. Here we have the cardiovascular outcomes. And you see a wonderful separation of the curves, 50% relative risk reduction. So what is this? Again, think of the pharmaceutical industry perspective. You go to see doctors and say, hey, look at this, look at the outcomes. Isn't that great? So, can you do it? Of course, you can do it, but what is the interpretation of this in this context?
That's effective.
Sorry?
That's effective.
It's effective, okay. So can you go and say evolocumab reduces cardiovascular events after this analysis?
It's difficult because, first of all, you do have the responders to the drug
Mhm.
included in the study, and what you're actually looking at here is it only responders to the drug, and it's a pre-specified exploratory
That's it. That's it.
analysis, so it's difficult to draw any.
Yep, absolutely. That's the point to make. This was an exploratory outcome and not the primary endpoint of the study. So this is, again, hypothesis generating. So this is how we have to look at it. It's very encouraging, obviously, but it's hypothesis generating. And that's obviously what was done. It was tested in a major outcome trial, and you know that's the FOURIER trial. So FOURIER, 27,000 patients randomized to evolocumab and placebo, and there was a significant benefit for the evolocumab arms. But that's exactly what needs to be done. So it's hypothesis generating, tested in an outcome trial, and that's what they did. Basel had a
I'm just going to ask you to go back to the OSLER second. Yes?
This one?
Please note, and this is very interesting, that they had a 53% hazard ratio 0.47 outcome. When they did the randomized trial, it was on the same endpoints, it was only 15%, 0.85.
Yeah, there you go.
Which is
Yes.
very educational. And
Yeah.
we see this in so many trials.
Yeah.
The meta-analyses or the pre-study prediction
Right.
is huge.
Right.
And when you saw the results, you go, oh, but it was also difficult.
Yeah. But it is very important. This is why we need randomized controlled trials. It's exactly this, okay? You have different magnitude of effect. In some cases, you have neutral. Sometimes you even have a negative effect. So that's why we actually need randomized controlled trials in the specific patient population powered for a certain primary endpoint. I couldn't agree more. So that's a wonderful example. Okay, so my last issue here. So this is something completely different. Now this is about a medical device. RESPECT was a trial that tested the efficacy of a PFO occluder, so patients with a cryptogenic stroke, or now we would say embolic stroke of unknown source, who had a persistent foramen ovale in the TEE study, were enrolled here, okay? Because you know that in these patients, clots may cross through the PFO from the right side to the left side of the atrium and then cause ischemic strokes. We know this. So basically, the RESPECT trial looked exactly at this. So cryptogenic stroke and PFO, and patients who had other explanations for a stroke, for example, carotid disease or atrial fibrillation, were excluded. Okay? So, and the endpoint was recurrence of non-fatal ischemic stroke or fatal stroke. Okay? So this was basically what was tested in the trial. So approximately 1,000 patients, randomization 1-to-1 to medical group, quite heterogeneous therapy, but one half got medical treatment, the other one was randomized to the device. So those of you who do interventional or may know about it, this is an Amplatz PFO occluder, which is simply placed in the interatrial septum to occlude the persistent foramen ovale. Okay, so this group received dual antiplatelet therapy, and then there was a TEE study done after 6 months. So very briefly, safety was excellent. Almost, nothing happened. Same rate for atrial fibrillation, no strokes during the procedure. Bleeding was identical. There were more vascular access complications because obviously if you randomize to medical treatment, you don't do anything with the patient. If you try to do a closure intervention, you need vascular access, which is done to the femoral vein, so you have to poke in patients, so you may cause some problems, and obviously there were more complications here. But other than that, everything was identical, so it was very safe. It was very successful. Technical success, meaning the occluder device is in the patient, very high, 99%. Procedural success was a definition within hospital. And then effective closure was evaluated 6 months after the study. So they did a TEE study, and they actually looked at residual shunting. So 93% of patients had an effective closure of their defect, high technical success, and a very safe procedure. Now, this is the outcome. As for all trials, intention to treat is there to test for superiority. So when you look at this, 50% relative risk reduction for the endpoint of stroke, okay? But look at the p-value 0.08, so not statistically significantly different. Look at the number of endpoints, 9 versus 16, okay? So a low number of endpoints, so I have to say that. So now here's the sad story about this. So to have a stroke because of PFO is very rare. So you have to say that. So it's one reason, but it's not very commonly happening. Obviously, every fourth of us has a PFO, so we're not all suffering strokes, right? So it's a rare event. Now, once you suffered one stroke, to have another one is even, it's also very rare. That's why neurologists will always say, let's go for medical treatment first and then wait, okay? And if it hits you a second time, then we can think about closure. So that's why. Now, what happened in the trial was that patients were randomized, and while waiting for the procedure, 3 patients in the device group suffered stroke. Okay, they were waiting to get the procedure, and in that waiting time, they suffered a stroke, which is very, very rare. So it's very unfortunate, but it happened, okay? So what does that mean? It means in the intention to treat analysis, patients were randomized, they're included in the analysis, right? So that's an event, that's a stroke. So basically it was counted as an event in the device group, although they never had a device. Okay, so when we look in the as-treated cohort, so these are the patients who actually got a closure device, you have a 70% risk reduction, significantly reduced, okay? So completely different interpretation. So let's say if that would have been intention to treat, to say, hey, this was a positive trial, but it was not, because intention to treat analysis was not significantly different. So then they put all this together. You see the relative risk reduction, 50%, 70%, number needed to treat went down over the years. It makes sense. So, but the interpretation was quite clear, technically. Of course, you have to say, neutral trial, there's no benefit, intention to treat. Okay, now we know that even after longer follow-up of these patients, there was a highly significant benefit. And this was just published last year in the New England Journal, together with 2 other trials, showing that there is a benefit, at least in certain patients with PFO closure. Okay, so now I have this. There were several meta-analyses, and I would like to show you this. So technically, there are different occluder systems, okay? So look at this one. This is the result for the Amplatz. So there were 3 different trials, I have to say, that testing this. 2 of them used the Amplatz device, and 1 used the so-called Starflex device. So when you look at this analysis, just looking for the Amplatz device, there was a significant benefit even in these trials. When you look for the Starflex device, it was not. Just look at this thing. This looks like, I don't know what to say to this. So I actually implanted these. They were terrible. So this is one thing. I also implanted these. They're very easy to handle from an interventional perspective. It's easy going, straightforward. So now look at this. Testing for heterogeneity. There's no heterogeneity. It's not, it's not different. So how can that be? Just look at these monsters here. How can there not be heterogeneity? Explain that to me, please. Why is that? Look at these beasts, they look completely different. Ugly, nasty beast here, and very smooth, easy going. So, why is that? You're not comparing the 2. What are you comparing? You're comparing a principle, same thing. Okay, you're comparing a principle, the principle of closure device versus medical treatment, and that's why there's no heterogeneity. Okay, but medically, it makes a big difference of what you put into a patient. Okay, and now we have this. That was even before the long follow-up of the RESPECT trial. So this was something looking, just comparing the intention to treat analyses of the trials, of the 3 trials, and looking for the as-treated analysis. And this is not statistically different. In the as-treated analysis, there was a significant benefit. So, and now comes my last slide, my question for you. You know, why should we use intention to treat in medical device trials? Or should we do as treated? Or should we maybe do something modified intention to treat? So what is your opinion? It's clear what is, the standard of care, the standard is intention to treat. Okay, but this is a perfect example why I think it's not the right way to go. So what should we do in the future? What do you think? There's no right or wrong answer here. Because I have a proposal for you, but what is your opinion? Both is always good. So you just choose what you want, right? Yeah. I mean, I think it's a perfect example why intention to treat, although it's the standard, it doesn't really make sense. That's my opinion. On the other hand, if you go for as treated, it means the patient has an occluder device in his chest and it did work out, right? So then he's counted in. You have to keep in mind that when you do a procedure like this, you have to poke a patient, you have to go in, you have to cross the septum, you're doing something, you can harm a patient by doing this. And this is safety, of course. And so I personally think it should be something like modified intention to treat, because you're attempting the closure at this moment, you're poking, you're going in, and maybe you fail because of a complication or you can't put the device, but that should be counted in because you're actually, you're doing something with the patient. So that's my personal opinion. It should be something like modified intention to treat, because then you have the patients with the device, but you're also covering the safety aspects of that. But that is not standard. This is just my personal opinion. Okay. I think this is my last slide. I told you we will do this 10 times. I think we did not. I think it was only 6 times. But here it is again. Okay. Join the working group. We are great people. Membership is free. And you can go through this link, which you will find on the webpage of our working group. Okay. Thank you very much.